All right, this is part 2 of the chapter 5 notes. You'll need your notes out so that you can write some things down. I'm going to start at page 57, so I'm going to go through a couple of things again. On page 57 and where it says passive transport, I don't think I really went over that very well, so I want to go ahead and go over that. Passive transport does not require any energy, and the reason it doesn't require any energy is because it there's diffusion down a concentration gradient. Concentration gradient is when something moves from a high to a low concentration. And so under the heading passive transport, doesn't require any energy. There's two types of passive transport. Simple diffusion and facilitated diffusion. Okay, so you have simple diffusion and facilitated diffusion. So I want to make sure everyone got that written down on page 57. You probably didn't get to fill in that blank. Two types of passive transport: simple diffusion and facilitated diffusion. All right, so I wanted to make sure I went through that. All right. Hypertonic, hypotonic, isotonic. I would have already gone through that in the Zoom call. Water potential. That is something that we would have already done in the Zoom call. We would have done problems on that by the time you're watching this. Hopefully you understand that. Water will move from a high water potential to a low water potential. Another way of saying that is low solute concentration to high solute concentration, and high pressure to low pressure. I would definitely just have those memorized, and then we're going to continue to talk about that as it relates to your egg and your potato, right? And then I would have gone over that. All right, let's talk about, we're going to start on page 58. Facilitated diffusion. So remember I said there's two types of passive transport. Passive transport meaning there no energy is required. The first type is just simple diffusion across the cell membrane. Osmosis is a type of simple diffusion across the cell membrane. Carbon dioxide moving from high to low concentration across the cell membrane is simple diffusion. Facilitated diffusion is the same concept, that something's moving from a high to a low concentration, except it's going to be one of those molecules that cannot freely pass the cell membrane, so it needs help from a protein. So that's the only difference. It's the same thing, same concept. It's just it's a type of molecule that needs help from a protein. So channel or carrier proteins are going to help hydrophilic substances cross that couldn't normally cross the cell membrane. It's like a door. So the substance is still moving from a high to a low concentration. It just needs a door to go through. It can't freely pass through the cell membrane. All right. So that's basically the only difference. So you could have a hydrophilic channel, or you can have what's called a carrier protein. So a hydrophilic channel loosely binds, carries molecules across, and so things that are going to need facilitated diffusion to move across are ions, polar molecules. Water, remember water can freely pass the cell membrane, but it kind of slows down a little bit. So if we want water to move really fast into or out of a cell, like with kidney cells, it's going to need a facilitated diffusion. All right, and then glucose is a sugar, which is very polar. It can't freely pass the cell membrane, so it's going to need a protein to help it. So it's going to need to do facilitated diffusion. All right, so facilitated diffusion underneath it on your page 58 says if a substance is hydrophilic, it may have problems getting across the hydrophobic region of the cell membrane. What helps achieve the transport of these substances, and draw a rough sketch? So I would draw a rough sketch of this. All right, and then I'd go ahead and pause right now and do that. All right. Does facilitated diffusion require energy? The answer is no, it still doesn't require energy because the substances are moving from a high to a low concentration. So equilibrium is being established. So no energy is required, but what is required is a helper protein to help these hydrophilic substances move. Our example of a channel protein is this thing called an aquaporin. An aquaporin is a channel protein that allows large quantities of water to pass through quicker and more freely than water would originally pass through the cell membrane. All right, so water can achieve simple diffusion, water also can achieve facilitated diffusion. When water does facilitated diffusion, it moves quicker. So an aquaporin is for water. It's found in large quantities in the kidneys. All right. A carrier protein. So a carrier protein is going to have a conformational shape change. So a conformational change. So what makes a carrier protein different from an aquaporin is that a carrier protein is usually going to help something that is much larger, like glucose and other sugars. And it will require a conformational change. So if you look right here, glucose is binding to a carrier protein. Once glucose binds to it, the carrier protein changes shape and then will spit glucose onto the inside of the cell. So that's the only difference. It's because glucose is so big, like you can't just have, so if you had a protein like this allowing glucose in, think about it. This isn't like a door that opens and closes. This is a door that's just always open. Glucose is so big compared to water, it needs a door that's closed. Like it's a doorway that's closed, then opens, then closes, then opens, then closes, then opens. Because glucose is so big. If glucose had a door that was always open, that would allow too many other things to pass through, if that makes sense. All right. So it's kind of like, okay, let me make an analogy here. So it's like a doggy door on your back door would be like an aquaporin. It's pretty much, well, it pretty much can be always open. Let's just act like, I don't know, the weather's okay. So if you had a doggy door on your back porch that was just always open, your dog could come in and out, and you don't have to worry about like burglars or anything like that. I mean, you might have to worry about some wild animals, but not burglars. I'm trying here. I'm just coming up with this off the top of my head. The glucose door would be like your front door where anyone could just come through if it was left open. So the doggy door, there's like a little flap. The dog can come in and out, no problems. Glucose transport's like your front door where too many big things like other humans that would rob you would come in and out. So we lock that door until glucose, which is like a key to open it, comes through. Man, I hope that made sense. These analogies that I just come up with on the top of my head, sometimes I think they confuse students more. I'll go over that again in class. I'll refine my analogy before we go to class next time. Okay, now we're going to switch gears here and talk about active transport. So active transport is the opposite of passive transport because it requires energy. Why does it require energy? Because it's going against the natural tendency of the universe to establish equilibrium, and it's almost going to be like an anti-equilibrium. It's going to cause even more differences across the cell membrane. So where passive transport is trying to achieve equalizing, on the flip side, active transport is doing the opposite of that. It wants it to be even more different. So I'm going to go over some examples. So the reason why active transport requires energy is because substances are moving against their concentration gradient. They're going from low to high, making it even higher on the other side and even lower on that one side. Our examples for active transport are going to be our electrogenic pumps. Our electrogenic pumps, in this class, we are talking about the sodium-potassium pump and the proton pump. A pump is just a protein that does active transport with ions. Okay? An electrogenic pump is going to generate voltage across a membrane. So if you took me during this summer, I've already talked about this. You actually have like very similar notes. So when I talk about the nervous system and neurons, I'm going to talk about the sodium-potassium pump. So if you already got that, awesome. It probably sucked at the time, but now you're glad that you did it. And then if you took me during cell bio, I went ahead and just gave this to you because I figured it would probably be a good idea. So if you've never seen electrogenic pumps before, please be sure to ask me and to make sure, because I don't know exactly how much emphasis the other cell bio teachers put on this. I put a lot of emphasis on it because it's a foundation for the central nervous system and the nervous system, period. All right, so electrogenic pumps create voltage across a membrane. What does that even mean? What does that even mean, voltage across a membrane? What that means is it's going to create a difference in charge on the outside compared to the inside. So the sodium-potassium pump and the proton pump are going to create voltage. They're going to create a difference in charge on the outside of the membrane compared to the inside of the membrane. The sodium-potassium pump is going to, and I would write this on the side, so these are your notes on page 59 on next to that picture. If you look at a step-by-step, so it's in a sequence, 1, 2, 3, 4, 5, 6, 1, 2, it's just going clockwise around. You see 3 sodiums get pumped out of the cell, 2 potassiums get pumped in. So what I say is there's 3 sodiums out for every 2 potassiums in. So how does that create voltage? Well, normally sodium and potassium aren't the only ions, and they're not the only charged things that are around. There's other ions like chlorine or chloride ions that are negative, right? And then you have proteins floating around that are very negative. So let's say the outside of the cell is -10, and the inside of the cell is also -10, okay? So if you take all of your positives, 3 positives out for every 2 positives in, do you see how it's then going to start becoming more positive on the outside? 3 positives out, and then you put only 2 positives back in. Over time, because the pump just keeps going and going and going, over time you end up with more positives on the outside than you have on the inside. And because they're also mixed with other negative things, it becomes more positive on the outside, more negative on the inside. This is a difficult concept. Students have a hard time with this, but it's something that you will need to get down. All right, so we'll talk about that a little bit more in the Zoom call, and I'll show you a little animation for it. All right, so that's the sodium potassium pump. What that does is it sets up the cell to want all of those positives to rush back in to achieve equilibrium. So if a gate opens, all of those positives rush back in, and it achieves work for the cell. So like the mechanical work of something rushing back in. Or just the voltage, the change of charge like propagates a signal along a neuron. It's a difficult concept. We will go over it more, trust me. But what I need you to get down right now is that an electrogenic pump is going to pump ions against their concentration gradient to generate a difference in charge on the outside of a membrane compared to the inside. And then I need you to know that the sodium-potassium pump releases 3 sodiums for every 2 sodiums out for every 2 potassiums in, and then that achieves that voltage across the membrane. I need you to know that a proton pump is going to push hydrogen ions against their concentration gradient, achieving voltage, and you can see it pretty good in this picture right here. Proton pump uses energy to pump hydrogens out against their concentration gradient. Well, hydrogens are positive. So then what that does is it achieves positive charges on the outside of the cell, and now there's a negative net charge on the inside of the cell. So then what do those hydrogens really want to do? They want to rush back into the cell. And so that sets up the cell to do work for cellular respiration, and it also is going to play a part in photosynthesis. Okay? So very important for y'all to know that. And then I'm going to play this video for you during the Zoom call, I think. Actually, maybe I should do it now. Hold on. All right, so you're going to want to pay close attention to this animation. There's no sound or anything. I'm going to just talk. So notice 3 sodiums bind. There's your energy. There's a conformational change. Those 3 sodiums then leave. They're on the outside now. 2 potassiums come in. There's a conformational change. those 2 potassiums leave. And it happens over and over. So watch it again. It takes ATP, 3 sodiums leave. Two potassiums come in. And it just keeps happening over and over again. So you can imagine at this point, there are many more sodium positive ions on the outside of the cell than there are potassium positive ions on the inside. So mixed with the negative charges, it is now going to be a net negative charge on the inside compared to the outside. So that is called voltage across a membrane. It's also called membrane potential. And I'll bring up that word again. Okay, good. I'm glad I got that done. Let me go ahead and go back because this is going to be less than 25 minutes because we're almost done. So I'm glad I got that done. You're going to definitely need to tell me at the end of this lecture on Edpuzzle if you did or did not understand it. Actually, I'm going to put it as a question on here because I need to know. All right, next type of active transport. So we had electrogenic pumps as our first type of active transport. Second type of active transport is it's going to combine a pump with facilitated diffusion. Okay, it has 2 parts. So co-transport has 2 parts. The membrane protein enables downhill diffusion of one solute to drive uphill transport of another. So let's just look at this picture here. So pause if you need to write that down because I'm about to explain this picture for you. All right, so this is called the sucrose hydrogen co-transporter. And this is a way that plants will do what's called sugar loading to help them like store sugar and whatnot. Sucrose is a carbohydrate, and sucrose is a disaccharide, as you can see in this little picture right here. So the cell wants sucrose to come in, but sucrose is just so big. It would just take so much work to get it in. And we have a better way to do it that is more efficient, that uses less energy than if sucrose were just to be brought in. All right, so hydrogens are pumped out against their concentration gradient using a proton pump and ATP. We now have a voltage across the membrane. It's a lot more positive on the outside of the membrane than it is on the inside. So what do hydrogens naturally want to do at this point? They want to rush back into the cell to achieve equilibrium, or an equal charge, an equal amount of hydrogen ions on both sides of the membrane. The only way that the co-transporter will let hydrogens come back in is if they bring a sucrose with them. It just has to do with the shape. Or what happens is hydrogen just comes back in, but then that opens up the door for sucrose because of the conformational change that happens to that protein. Okay, so you see how this is combining a proton pump with what's called a co-transporter. So as hydrogen comes back in, there's a conformational change in the protein that now allows sucrose, and it's just more energy efficient to do it that way. Okay, I want to go ahead and you're going to make a T-chart that compares passive to active transport. So go ahead, pause the video, and write this down. We're just going to compare the two. Passive transport requires no energy, and let's just say no energy. Things are moving from a high to a low concentration, down a concentration gradient, and here are your examples. Diffusion, osmosis. So diffusion, I call it simple diffusion. Osmosis is the simple diffusion of water. Facilitated diffusion is diffusion that requires a help with some kind of transport protein, whether it's a protein channel or whether it's a carrier protein. All right, so this is just your summary of everything we just talked about. Active transport does require energy, and the reason why is because it is moving things, most likely ions, from a low to a high concentration. So they're moving against their concentration gradient. And bulk transport like endocytosis and exocytosis. So we talked about pumps already, electrogenic pumps. Your other example of active transport, so we talked about electrogenic pumps. We talked about the sodium potassium pump. We talked about the proton pump. We talked about the sucrose co-transporter. A co-transporter can be used for any sugar, by the way. Now I want to tell you about endocytosis and exocytosis, our last type. And there's a picture. It's a really good picture to summarize what we just talked about. I don't want to talk about that just yet. So bulk transport is it's a type of active transport that uses the cell membrane. And it's just the use of vesicles. So we basically already talked about vesicles when we talked about the Golgi apparatus and the endoplasmic reticulum. That requires ATP to do that. So it's the use of the membrane and vesicles to transport things that are just way too large. I mean, no point in even trying to do a channel protein or a carrier protein. They're just too big and in too large amounts. So just bring them in with a vesicle or push them out using a vesicle. All right, so endocytosis takes in macromolecules. Exocytosis is going to free. So endo brings in using the membrane and a vesicle. Exocytosis dispels them or secretes them, whatever word you want to use. Pushes them out using a vesicle and the membrane. See how the vesicle fuses, releases them. It's pretty cool. So that requires ATP. There's 3 types of endocytosis. Phago is cellular eating, pino is cellular drinking, and then receptor mediated. So phagocytosis, you're going to want to write down the examples. Phagocytosis would be like food particles, but I think a really good example for phagocytosis is a white blood cell engulfing a bacteria. Pinocytosis is cellular drinking, when the cell just wants to bring in a large amount of sugars and ions at one time, and it doesn't want to use a channel protein to do it. It's going to take in just a bunch of the liquid, and it's just going to have a lot of sugars and like little proteins in it and stuff like that. So it's not like one big giant solid. It's just a bunch of little molecules coming in. And it's called cellular drinking because it's a mostly fluid that's being brought with it. Receptor-mediated endocytosis is when it's more specific. Receptor-mediated endocytosis is just like pinocytosis, except it's very specific about what it wants. So instead of just engulfing a large amount of fluid and just hoping you get what you want coming in, this one, a sugar or a protein is going to have to fit a receptor. It's like a lock, a key that fits a lock. Okay, yes, this is the actual protein I want to bring in. Then it's going to bring it in. All right. Receptor-mediated endocytosis is also how viruses enter into a cell. So a virus will mutate so that it has a protein that matches a receptor. It's going to trick the cell into thinking, oh yeah, okay, this is what I was looking for. And then it comes in and it like uncoats itself, and then it infects the cell. All right. A ligand is something that binds to a receptor. So that's a word that you're going to need to know. A ligand is something that binds to a receptor. So if it's a protein that's trying to match with a receptor, like unlock, it's a key that unlocks a door, like an entry of coming in through a vesicle. The ligand is the thing that matches the receptor. All right, and then that's really all I have for you with that. I think one thing though, we're just under 25 minutes, so let me, I'll talk about osmoregulation during class. Let me just do that during class. All right, I'll talk to you guys later.